Okay, so this is another short clip. Again, bear with my videography, so many technical issues. But this is the part three from where I ended, where I was describing just the trophozoite that you will appreciate in the microscopy, that you did appreciate in the microscopy. Okay, so the one last part before I actually go through the procedure quickly of the count. What was mentioned in the introductory note was, remember that your parasite resides in the red blood cell, the erythrocytic stage. And so the specimen is a blood specimen, and therefore we are making a blood smear for the microscopy that we are using as a tool. So I quickly run you through the two very important types of smears that are made, which was a thick smear and a thin smear. Okay, so as the names say, a thick blood smear is made is made to be thick, and the thin smear has to be made in such a way that it is a thin impression. Okay, so we are using just 2 drops of blood on the thick, so that we are able to make that circular impression that is thick. And then on the other side, on the thin, we use 1 drop of blood, and then we spread that across on the clean slide to make a thin impression. But the most important distinction you saw for yourself in the microscope is that you have a preservation of red blood cells in the thin smear because of the way it is prepared, where we fix the slide in alcohol. Okay, and that step preserves the red blood cells under your thin smear, so we're able to see the parasite inside your red blood cell. On the other side, on the thick smear, we do not have red blood cells present anymore. And so when you are seeing it under the microscope, you are just seeing your slide, the GEMSA stain, of course, as the background, and then if the parasite was present, just simply suspended in the slide, okay, and some white blood cells, okay. So that's about it, really, with the types, and now we can consolidate on how we use these types of smears to do an actual count, okay? So in the session, I explained that there are 3 types that I use, plus grading system, percentage parasitemia, and parasites per microliter, okay? So I have the chart that I used from the session. So basically, the plus grading system is almost it's not as popular, like I said, in the lab, but it's still relevant. So you can go through and study it, okay, and see what it means when you hear you have a +1 malaria, you have a +2 malaria, and so on. But that whole grading system is generated from a thick smear. Okay, so but the most informative, because they really give you a good sense of the infection, are the percentage parasitemia, as you can see on the board there, and parasites per microliter. So you had parasites per microliter, your thick smear, as task 3, okay, and then task 4 was percentage parasitemia up there, okay. So basically the task, for example, under your thick smear, you're supposed to generate a counting field, appreciate the microscopy, okay, when we're about to count using a thick smear. So you drew something like this, identify if there are parasites inside and some white blood cells, and then we have the chart itself, the tally, how we generate the data that was already supplied to you. So I have something like this that I explained already in the session, but basically you start in whatever field, your starting point, field 1, and then you document how many in that field, how many parasites are in there, you document that, you record how many white blood cells in the same field, you count that as well, and then you go to the next field and do the same. Document the parasites and also the white blood cells. And you continue this process up to the cumulative total of 200 white blood cells, and you stop, and then you see how many parasites are in 200 white blood cells. and you feed into that formula. Okay, that formula we were given in class, in the session, okay, and I explained how to use it, okay, where this value is coming from and so on. Okay, so please apply yourself with the data provided, and then also after that calculation you have to comment on the disease severity. Remember that that is part of task 3. Okay, disease severity according to WHO reference, very important. And then finally, percentage parasitemia. Okay, just a quick wrap up here. Percentage parasitemia, I hope you can see. So we have a different tally according to what is actually preserved and the counting elements available in the slide, okay? So you have a tally of infected red blood cell and overall number of red blood cells, okay? So you have a little sketch here that I did. For example, your field 1, you do a count. How many in this field, how many red blood cells are infected? You document that, okay? And then also, you document overall how many red blood cells are present, and you document that as well. So whether infected or not infected, you document that. And then you go on to the next field. You do the same, field 2, and so on. And it's usually, by the time you reach field 4, you will have a total of about 500 red blood cells, okay, usually because they're numerous in each field. Okay, so by field 4, you have reached your 500 or so, you stop, okay, and then see how many, the proportion, okay, how many infected red blood cells are in the total 500, okay, and then you feed into that formula, okay, that you can see up there, okay, something like that. And then again, of course, this is task 4, so you are